every second. Your body is running 37 trillion operations, but right now, one molecule is about to hijack all of them. A clear liquid sits in a glass, deceptively simple. Tito's handmade vodka, 40% ethanol, 60% water, nothing else. No fat, no protein, no carbohydrates. Just one molecule, C2H5OH, 2 carbon atoms, 6 hydrogen, 1 oxygen. This molecule was born from corn. Saccharomyces cerevisiae. Microscopic yeast organisms consumed the corn's glucose and exhaled ethanol as a byproduct. A fermentation process unchanged for 10,000 years. Then distilled, filtered through charcoal. Each pass stripping out congeners, impurities, everything except the ethanol and water. Then bottled. Then poured, and now it begins. The moment ethanol contacts the oral mucosa, the body responds. TRPV1 nociceptors, the same nerve endings that fire when you touch an open flame, activate immediately. That familiar burn is not flavor. It is a pain signal, a biological alarm. Chemical detected, warning issued. The 3 salivary glands, parotid, submandibular, sublingual, flood the mouth with saliva. Amylase enzymes deploy, ready to digest starch. But vodka carries no starch. The enzymes float, useless, finding no substrate. Because ethanol needs no digestion, it requires no enzyme, no transporter, no energy. Even before swallowing, the buccal mucosa, the thin lining of the inner cheek, begins absorbing ethanol directly into the submucosal capillaries. The journey has begun before a single conscious decision to swallow has been made. The epiglottis seals the airway with biomechanical precision. The pharyngeal constrictor muscles, superior, middle, inferior, fire in coordinated sequential waves. A peristaltic escalator propels the liquid downward at 3 to 4 centimeters per second through 25 centimeters of muscular esophageal tube. The esophageal lining, stratified squamous epithelium, has no thick mucus protection, no armor against chemical assault. Ethanol touches it directly. TRPV1 nociceptors fire in the submucosa. The lower esophageal sphincter, weakened by ethanol's smooth muscle relaxation effect, opens. The vodka enters the stomach, and the lower esophageal sphincter, that critical anti-reflux gate, remains inappropriately relaxed. Acid reflux risk has already begun. The rugae folds smooth, the stomach expands, and the gastric machinery activates with extraordinary speed. Parietal cells engage the H+/K+AT-pase proton pump, one of the most powerful molecular machines in human physiology, driving hydrogen ions into the gastric lumen. pH crashes to 1.5, an environment capable of dissolving metal. Chief cells release pepsinogen, active into pepsin. G cells secrete gastrin, the full digestive arsenal, mobilized for liquid requiring none of it. Because ethanol bypasses digestion entirely. Through passive diffusion across the simple columnar gastric epithelium, ethanol crosses directly into submucosal capillaries. No enzymatic processing required. The gastric alcohol dehydrogenase enzyme, stationed as the stomach's last chemical defense, neutralizes only 10 to 20% of the ethanol load. The remaining 80% enters the portal venous system intact. But the stomach pays a price. The protective mucus gel layer thins under ethanol's chemical assault. Mast cells degranulate, releasing histamine. Prostaglandin synthesis is suppressed, removing the stomach's primary mucosal repair mechanism. Neutrophils begin infiltrating the lamina propria. The stomach lining is inflamed. After one glass, the stomach is already at war. The pyloric sphincter releases controlled spurts of chyme into the duodenum. The body responds with its full digestive complement. The pancreas releases lipase, amylase, trypsin, chymotrypsin into the duodenal lumen. The gallbladder contracts. Cholecystokinin triggered, releasing bile, golden green, rich in bile salts ready to emulsify fat. But vodka contains no fat, no protein, no starch. These extraordinary molecular machines, each one a precision evolutionary product, have no substrate to act upon. They float enzymatically idle in a lumen that offers them nothing to process, because ethanol requires zero enzymatic digestion, already the perfect size, already water-soluble and lipid-soluble simultaneously. It passes the brush border enzymes completely unimpeded, sucrase, lactase, peptidases all ignored, and absorbs directly through the enterocyte lipid bilayer membrane via transcellular passive diffusion. The jejunum, with its plicae circulus folds, its towering villi, its microvilli brush border, amplifying absorptive surface area 200-fold to the equivalent of a tennis court, absorbs ethanol at maximum biological rate through the enterocytes across the fenestrated submucosal capillary endothelium into the portal blood. The mesenteric venous tributaries converge. The superior mesenteric vein collects the ethanol-enriched blood. The portal vein forms, carrying its concentrated ethanol payload directly to the liver's porta hepatis. The liver lobule receives the ethanol-laden portal blood through 6 portal triads simultaneously. It flows through the hepatic sinusoids, past Kupffer cells, the liver's resident macrophages scanning every molecule, through the fenestrated sinusoidal endothelium, across the space of DC and into the hepatocytes. Inside zone 3 centrilobular hepatocytes, the cells bearing the heaviest ethanol exposure, the ADH pathway activates. Alcohol dehydrogenase. A zinc-containing metalloenzyme binds ethanol in its active site. NAD plus accepts a hydride ion transfer from ethanol. The reaction produces NADH and acetaldehyde. Acetaldehyde. This is the true toxin, not ethanol. Acetaldehyde. This reactive aldehyde is responsible for every classic intoxication symptom: flushing, headache, nausea, tachycardia. It forms protein adducts, damages mitochondrial membranes, initiates inflammatory cascades. In the mitochondrial matrix, ALDH2, aldehyde dehydrogenase, converts acetaldehyde to acetate, relatively benign, released into systemic circulation for peripheral metabolism. But the metabolic cost is severe. NAD accumulates, overwhelming the NAD plus pool. The elevated NADH/NAD plus ratio blocks gluconeogenesis. The liver can no longer produce glucose. Hypoglycemia risk rises. Beta oxidation of fatty acids is inhibited. Triglyceride synthesis accelerates. Lipid droplets, macrovesicular fat deposits, begin forming inside hepatocytes. The smooth endoplasmic reticulum's CYP2E1 enzyme generates reactive oxygen species, superoxide, hydrogen peroxide, hydroxyl radicals, attacking lipid membranes, oxidizing proteins, damaging DNA. After one drinking session, fatty liver has already begun. The liver processes one standard drink per hour, no faster. Everything beyond that flows directly to the brain, the blood-brain barrier, the most fortified biological boundary in the human body. Tight junction proteins, claudin 5-occludin, form an almost impenetrable paracellular seal. Most molecules are blocked. Ethanol, small, amphipathic, lipid-soluble, crosses the endothelial cell membrane via transcellular diffusion without resistance. Inside the cortical neuropil, ethanol acts simultaneously on two opposing receptor systems. First, GABAA receptors, the brain's primary inhibitory ionotropic receptors. Ethanol binds allosteric transmembrane sites, potentiating chloride channel opening. Enhanced chloride influx hyperpolarizes neurons. Inhibitory tone increases throughout the cortex, limbic system, cerebellum. Anxiety suppressed. Behavioral inhibition reduced. Motor coordination impaired as Purkinje's cell function in the cerebellum degrades. Second, NMDA receptors, the brain's primary excitatory receptors. Ethanol blocks the channel pore, preventing calcium entry. LTP, long-term potentiation, the cellular mechanism of memory formation, is inhibited. Learning capacity is suppressed. Judgment, housed in the prefrontal cortex, begins to erode. And then, the mesolimbic reward pathway activates. In the ventral tegmental area, GABAergic inhibitory interneurons are silenced by ethanol's GABAA potentiation. Dopaminergic VTA neurons released from inhibitory control fire rapidly. Dopamine floods the nucleus accumbens. Serotonin releases from the Rafi nuclei. Beta endorphins release from the hypothalamus. The subjective experience: euphoria, confidence, social ease, pleasure. This is the neurochemical architecture of why alcohol consumption has persisted across every human civilization in recorded history. But the mechanism is a chemical deception. The brain is signaling reward when no adaptive behavior has occurred. With repeated exposure, the mesolimbic pathway reorganizes. Tolerance develops. Dependence follows. Simultaneously, ethanol distributes to every organ through the systemic circulation. The heart. Cardiac myocytes experience altered calcium channel kinetics. The sinoatrial node accelerates. Heart rate increases. Peripheral vasodilation occurs via endothelial nitric oxide release. Blood pressure falls slightly. The skin flushes. The sensation of warmth is thermoregulatory deception. Heat is being lost to the environment through dilated cutaneous vessels, not gained. Skeletal muscle. Mitochondria preferentially oxidize acetate from hepatic ethanol metabolism, displacing fatty acid beta oxidation. mTORC1 signaling, the master regulator of muscle protein synthesis, is suppressed via ethanol-activated inflammatory kinases JNK and IKK beta. Muscle protein synthesis falls. Glycogen resynthesis is impaired. Bone. Osteoblast activity is suppressed under ethanol exposure. Osteoclast activity continues unimpaired. The net result: bone resorption exceeds formation. Vitamin D activation is reduced in the ethanol processing liver. Calcium absorption falls. Bone mineral density declines, quietly, invisibly, with each exposure. The gut microbiome. Beneficial lactobacillus and bifidobacterium species decline. Pathogenic gram-negative proteobacteria expand. Tight junction proteins. Claudin, occludin, ZO1 disorganize from their paracellular positions. The intestinal barrier becomes permeable. Bacterial lipopolysaccharide crosses into the portal and systemic circulation. Kupffer cells in the liver bind LPS via TLR4 receptors, activating NFKB, releasing TNF-alpha, Illinois 6, and IL-1 beta into systemic circulation. A quiet, invisible systemic inflammatory fire ignites. The kidneys. Ethanol suppresses vasopressin release from the posterior pituitary. Without ADH signaling, aquaporin 2 water channels fail to insert into collecting duct apical membranes. Water passes into urine unabsorbed. Urinary volume increases dramatically. Magnesium, zinc, and potassium are excreted in excess. Dehydration develops, even as fluid is being consumed. But the body never stops fighting. As blood ethanol concentration begins to fall, the clearing operations begin. The large intestinal colonocytes absorb residual water through AQP3 and AQP4 channels. The gut microbiome initiates slow ecological recovery. Liver cool crypt stem cells divide, producing replacement enterocytes to repopulate the ethanol-damaged intestinal surface. The posterior pituitary restores vasopressin secretion. Aquaporin 2 channels reinsert into collecting duct membranes. Water reabsorption resumes. Electrolyte homeostasis begins restoring. The lungs exhale ethanol vapor with every breath. Henry's law establishing the 2-1-0-0, 1 blood to alveolar partition ratio that breathalyzers exploit for back estimation. The liver continues its steady enzymatic work, 1 drink per hour. ADH oxidizes ethanol to acetaldehyde. ALDH2 converts acetaldehyde to acetate. Peripheral tissues, heart, skeletal muscle, brain metabolize acetate as fuel. The BSE curve descends at 0.015 to 0.02% per hour. The GABAA potentiation fades. NMDA receptors recover, sometimes overshooting baseline excitability, producing the restless, anxious morning after rebound. The dopaminergic reward signal dims. The body comes back, cell by cell, system by system, not without cost, but it comes back because that is what this body does every single time without being asked. When you raise that glass, 96 biological events mobilized, a mouth firing pain signals, a stomach dissolving its own protective lining, a liver converting poison to acetaldehyde, a brain rewiring its reward architecture, a heart changing its rhythm, muscles losing their synthetic capacity, bones losing their density, a microbiome losing its balance, kidneys losing their ability to retain water, every organ, every cell, every molecular pathway, responding, processing, protecting, repairing, silently, without complaint, without recognition. Your body has been fighting for you your entire life, without a single request, without a single thank you. The least it deserves is to be understood. If this documentary changed how you see what happens inside you, share it. Because the most powerful thing science can do is make the invisible impossible to ignore. 96 battles, one body, now you know.